Dr. Madangi Ganapati has obtained her MSc in Genetics and Plant Breeding from Kerala University and PhD from School of Biotechnology, Madurai Kamaraj University. Having a passion for gaining knowledge and for instilling curiosity in fertile minds, she has been active in the field of teaching and research for more than 15 years. Her current research interest includes recombinant protein expression in plants, transgenic plant development and manipulation using molecular techniques, and analysis of changes in basic gene regulatory pathways during transgene expressions, nutraceuticals, and the role in alleviating certain chronic human diseases. She has a number of publications in peer-reviewed international and national journals.
Welcome to the lecture series on food microbiology. When we trace the evolution of food, this is an integral part of humankind, and the ancestral evolution says that mankind started as nomads, hunters and foragers, later on settling down in a place for agriculture. The moment man started agriculture, he had to start thinking about how to preserve the products which he could make, because climatic conditions are always not conducive. So between the 8000 and the 1000 BC, you had many preservation techniques such as drying, cooking, smoking, then preserving the food using salt, sugar. Then you had the low temperature storage of grains. Then you have history saying that people used to store the grains in mud pits, which is storage without air. Then fermentation, pickling, spicing, etc. Fermentation was used not only for preservation, but it was also used for increasing the nutrition factor to produce a distinct flavor, etc. And if you trace the history of food microbiology, it was Person in 1822 who first found there was a microscopic organism which was found on the surface of wine, which he called as Mycoderma mesentericum. And Louis Pasteur in 1868, he said that this particular organism was associated with the conversion of alcohol into acetic acid, and he called it as Mycoderma acetae. And in 1898, Bejernik renamed this as Azotobacter aceti. And you had it in 1837, Schwann named the organism as Saccharomyces, otherwise called as a sugar fungus. And in 1860, Pasteur showed that the fermentation of lactic acid and alcohol from sugar was the result of the growth of specific bacteria and yeast. And then in 1883, you had Christian Hansen, who used the pure cultures of yeast for fermentation. This knowledge was the beginning of the separate field of food microbiology. Once the role of food and microbes, association of microbes with food was known, scientists started investigating the role of microbes in food industry, especially the microbes which can be used to preserve, to increase the nutrition factor of food, prevent the growth of other microbes, especially the pathogenic species in food. And also, the another important factor was the growth of certain microbes in food could cause allergenicity or toxicity. So this also became an important field. In 1820, you had Justin Kerner, who observed that food poisoning could be caused by eating blood sausage. Here it was Clostridium botulinum which was the culprit. And in 1849, you had John Snow, who suggested the spread of cholera by drinking contaminated water, especially water which is contaminated with sewage. And in 1864, the cholera bacilli was first discovered. It was Filippo Fazzini who called it as Vibrio cholera, and later on Robert Koch identified it and isolated it in pure culture. And in 1885, Theodore Escherich isolated a bacterium coli, which was later called as E. coli or Escherichia coli, from the feces, and suggested that the presence of these strains in some food could be the cause of infantile diarrhea. And all these developments led on to the major development in the field. Before going in depth into the field of food microbiology, let us first have a recap on general microbiology. Now let us see the connection between food and microbes. In general, what do you call as microbes? You have the bacteria, you have the fungi which consist of the yeast and the mold, you have the protozoa, you have the microscopic algae, you have the viruses, even the parasitic worms come under the class of microbiology. Now, how does a human being get exposed to this? The first is ingestion, that is you eat, you consume, either drink water or eat food containing these microbes. Second is injection. either wantedly or unwantedly you get exposed to the organisms by needles. The third is inhalation, that is you along with air, when you inhale the air along with it you get exposed to these microorganisms. So these are the three major modes or routes of exposure of microbes to humankind. Generally when we call as microbes, it means that they are too small, normally too small to be visible. You see them only with the help of microscope. They can be either swimming freely or they might be attached to the substratum. There are organisms which form biofilms, and when you have different species or different types of microbes associated with a different sphere within a place, you have interaction between these communities. As a result of this interaction, it can be harmful to humankind because the interaction can produce waste products which can be toxic or noxious, or in some cases it can be beneficial because what one organism produces as a waste and which is toxin can be taken up by another organism as its energy source, thereby that toxicity is reduced and the waste gets converted into a nutrient. So microbes can be classified. We can say that they can be useful, in some cases it is beneficial, or they can also be toxic. Now you have organisms which are pathogenic, which is disease causing. There are many organisms which are capable of decomposing the organic waste. They are the organisms which can do photosynthesis. You have the typical purple sulfur bacteria. For them, they fix atmospheric carbon dioxide. You have many organisms which play an industrial role in fermentation to produce ethanol, acetone, alcohol, and so many other products. Then among the food industry, for bread, cheese, vinegar, and so many other fermented food products, we use microbes. And again, for breaking down cellulose, we have the cellulase producing organisms. Then nowadays, with the advent of genetic engineering, we have converted the normal organisms into those which are capable of producing insulin and other therapeutic products. So if you see, if we categorize these organisms, now yeast and lactobacilli, these are major organisms which are commonly used in food industry. Then you have organisms which are used for wastewater treatment, example the bacillus species. You have many organisms which can actually decompose or digest the matter which is present in the waste products, and these actually, the advantage here is they convert it into a waste which is not toxin without producing any noxious fumes or toxic matter, toxic to humankind or any other living organism. You have many organisms which are capable of decomposing organic and inorganic compounds and producing methane, biofuels, etc. But in science we use them for laboratory experiments, and the other major aspect is biowarfare, where the potential virulent organisms are used as weapons, bioweapons. A typical example is the organism causing anthrax, which is Bacillus anthracis. Now, if we talk about fermentation and the microbes which are associated with it, a typical example is tempeh, salted fish, etc. Here, fermentation, the microbes which ferment not only increase the flavor of the food, but they also help in preserving the food for a longer time. And then you have the decomposition for the production of natural gas, ethanol, biofuels, etc. Then you have microbes which are used in clearing up oil spreads. For example, we call it as leaching. I am sure you have heard about it, that when ships and containers carrying petroleum products break down in the oceans, we have the recombinant microbes which are sprayed in those areas to break down the oil spill leach. This is done by genetic engineering methods. And you have also organisms which are capable for removing the heavy metals and toxic components from soil and other areas and converting them. We call this as bioremediation. Now, that is one aspect. Then you have the other aspect, E. coli, which is actually a common dweller in the gut microbiota. It's a gut microbiota, meaning all human beings have E. coli. It is present all over. But you have cases where this normal human non-pathogenic organism which is capable of producing diarrhea. So then you have the HIV, which is very known, human immunodeficiency virus. You have an organism which causes the ulceration. the herpes viruses which cause the pox diseases. Then you have the malarial parasite, the common cold or the influenza virus which exists in different types of strains. Then you have the Ebola, which is now famous because this is one where we have now synthesized some sort of vaccines for it, but this is highly contagious. So you have so many microbes which are pathogenic, which can cause multiple diseases. So let us see the basic classification of microorganisms. Basically, the domain is divided into bacteria, archaea and eukarya. Now, what we call as bacteria and archaea together comprise the fungi, the protozoa, the algae, the viruses and even the multicellular animal parasites, by which I mean the helminths. You have the ascaris, the roundworm, the hookworm, the tapeworm. All these also belong under this class of disease causing organisms. Among the eukarya, you have the plants, animals, fungi and the protista. Let us take and consider each class of these organisms. If we take archaea, they are prokaryotic. Now, why is this archaea separate from bacteria? That's a basic question. Why can't you club them together? What makes them so distinct that they require a separate domain? They are prokaryotic, but the basic difference is that you know that most of the cells have a cell wall. In bacteria, this cell wall is comprised of a material which is called as peptidoglycan. And these archaea, the microorganisms which belong to archaea, do not contain peptidoglycan. The second difference is that they live in extreme conditions, meaning in temperatures, that is, extreme thermophiles come under them. They are capable of living in temperatures which are very high. Then extreme halophiles are there which live in high alkaline solutions. Then you have the methanogens. So they are distinct. They have a distinct environment in which these archaea can survive. Bacteria, these also belong to prokaryotes. The difference between the bacteria and the archaea is that bacteria have a peptidoglycan cell wall. As you see in the picture, you have the different types of bacteria which we have represented here. See, here you see an endospore. Here you have two round shaped organisms which are stuck together. So you have a spiral shaped organism here. Here you have an organism with so many cilia, flagella which are around. This has only flagella towards one side. So different types of organisms are present in the bacteria. We will be talking more about that later. Now, when we talk about this bacteria as a class or a domain, they can be phototrophs, that is autotrophs, or heterotrophs, meaning many utilize organic and inorganic chemicals to synthesize their food, or many can do photosynthesis to obtain energy. Then we come to the class protozoa. A very well known example of this is amoeba. This is a unicellular eukaryote. They normally absorb or ingest organic chemicals. Now, what is the difference is that here, in the case of a bacterium, it cannot change its shape randomly. But in the case of these amoeba-like organisms, the protozoa, these extensions are called as pseudopodia, meaning their cell wall, when they move along in search of food, these extensions can be formed in the area. The cell wall can extend towards that area where food is available. That is why it's called as pseudo, because false. It is not actually something which is real, which is rigid, but something which can change its position for locomotion. So they have either pseudopods, cilia or flagella, and a typical example of this is the amoeba. Then you have algae. As you all know, you have unicellular algae and multicellular algae. These have cellulosic cell walls. They are the precursors of the present day plants. They have cellulosic cell walls. Almost all of them gain energy through photosynthesis, so they have photosynthetic pigments. So red algae, blue-green algae, green algae, brown algae. So depending upon the type of pigmentation available present in them, or if you would say the type of pigments which can trap energy from the sunlight to do photosynthesis, we name them as different classes. They can produce molecular and organic compounds. Fungi belong to Eukaryota. Here you have chitin cell wall. and they use organic chemicals for energy. And these we normally divide into yeast and molds. Okay, yeasts are unicellular, but molds and mushrooms are multicellular, and they compose of what we call as mycelia, which are composed of filaments which are called as hyphae. So you have yeasts here in the class of fungi. You also have molds and mushrooms, which are mostly decomposers. Now we come to the next major class, the viruses. They are even smaller than bacterium, normally are visible under the electron microscope. If you see the genetic material, it can be DNA or RNA. In all the other classes of microbes which I so far said, your algae, the bacteria, the fungi, in all of them the genetic material is composed of only DNA. But viruses are the one class where you can have a genetic material which is either DNA or RNA. The other distinction is that the virus is normally genetic material DNA, which is present in eukaryotes, is double stranded. It doesn't exist as a single strand, as you all know. But here in the case of viruses, you can have the genetic material divided into a single stranded DNA, a double stranded DNA, a single stranded RNA, or a double stranded RNA. So depending upon the type of genetic material which is available inside, you can divide the viruses class into different classes: single stranded RNA viruses, double stranded RNA viruses, single stranded DNA viruses, and double stranded DNA viruses. Now the structure of a virus is actually very simple. It just has the genetic material covered by a protein coat, and some do have what you call as a lipid envelope outside. Okay, so we now call them as either enveloped viruses or naked viruses, depending upon whether they have a lipid coating envelope outside or whether they don't have, meaning their structure is very simple, just genetic material with a protein coat. And as you all know, viruses are not alive outside a living organism. So for them to be alive and replicate, they need a host cell. Outside a host cell, they don't have an existence. But are they present everywhere? Yes. So they are replicated only once they are inside a host cell. And most of the viruses are specific in their host. So you have organisms, the viruses which will infect only bacterial cells. We call them as bacteriophages. Then you have specific plant viruses, they infect only the plants. Then you have the animal viruses. And you also have what you call as viroids, which are nucleic acid, there is no protein coat. And then you have the other case, the other extreme, prions. They are infectious protein particles, and they are the causative organisms for what you call as bovine spongy encephalitics, or what you call as a mad cow disease. So all these, which are too small to be visible even under the light microscope, they are extremely infectious. Let us see the modern prokaryotic classification. We classify them as either eubacteria or archaebacteria, the cyanobacteria, and the thermophiles. And these slide shows the different families which are involved which are classified in each of these archaebacteria and the eubacterial species. Let's take archaebacteria. These archaebacteria do not have any cell nucleus, okay, and there is no membrane bound organelle within their cells. So earlier they were clubbed under bacteria, but they were supposed to be classified as an unusual group of bacteria, mainly because no cell nucleus, no membrane bound organelles, and then called as archaebacteria. And now what has happened is that we already know that archaea has an independent evolutionary pathway and independent evolutionary history, and if you trace the biochemical pathways, these archaea have separate metabolic pathways which are distinct from bacteria. Okay, so now we classify these archaebacteria into a separate domain. Let's talk about bacteria now. The bacteria basically can be classified based on their shape, based upon the relationship with how they accept certain dyes and what is visible, and based on the oxygen demand. So based on shape, you have the spirillum, the spirilla, you have the bacilli, you have the cocci. Based on their ability to accept gram stain, you have the gram positive and gram negative. This depends upon the thickness of the peproglycan layer of the cell wall, as you are all aware. Then you have based on oxygen demand, that is whether they require oxygen for growth, then they are aerobic. If they do not require oxygen for growth, they are anaerobic. Now, so based on the nutrition types, what is the energy source? So, if chemical compounds are the source of energy for the organism, they are the chemotrophs. And if light is the source of energy, they are the phototrophs. So, you have based on the energy source either chemotrophs or phototrophs. Now, based on the carbon source, we can again subdivide them. Is the carbon source an organic compound or is the carbon source carbon dioxide? So, if it is a chemotroph and then it is utilizing organic compound for its carbon source, it is a chemoheterotroph. And if it is using carbon dioxide as the carbon source, it is a chemoautotroph. Now, this chemoheterotroph can again be divided based on what is the final electron acceptor molecule. If it is oxygen, for example, all animals, most of the fungi come under this class. If it is you have then classification. If it is not oxygen, then depending upon whether it is an organic compound or an inorganic compound, you have the fermentative bacteria, or in the case of inorganic compound, whether it is accepted in the electron transport chain, for example, the Clostridium. Now, if carbon dioxide is the carbon source, you have a major classes of organisms like your hydrogen bacteria, hydrogen sulfur bacteria, then iron cores can be nitrogen. So, like that the classification differs. Now, let us take the other class where you have light as the energy source. These are the phototrophs. Now, divide them into the carbon source. Again, you have the carbon organic compounds, you have carbon dioxide as the carbon source. So, we divide them as the photoheterotrophs and the photosynthetic or the photoautotrophs. Among the photoheterotrophs, you have the green non-sulfur bacteria, you have the purple non-sulfur bacteria, etc. Now, if these photoautotrophs utilize water to reduce carbon dioxide, that is a question. So, now if the answer is yes, then the type of photosynthesis which is happening is oxygenic photosynthesis. Your plants, cyanobacteria, come under this class. If they do not utilize, then they are anoxygenic photosynthetic organisms. Green and purple bacteria will come under this class. So, what is the mode of nutrition? What is the energy source? What is the carbon source? And what is the final electron acceptor? So, based on these categories, we can classify microbes, bacteria. So, basically we divide it by the shape, as I said, spirilla, bacilli, cocci, then ability to retain the dye, we say that gram staining, then that is the major identification factor, is it gram positive or gram negative. There are certain microbes which do not accept gram stain, then you can actually use other staining methods like acid fast acid fast staining, etc., to classify them, to identify those organisms. Then whether you require oxygen, ability to grow with air or without air, then the final classification would be based on biochemical reactions, what are the different metabolic pathways. So, if you are given a specimen and you are asked to identify what is the bacteria, you first look at its shape under the microscope, then you do a gram staining to identify whether it is gram negative or positive. You do a series of culture, keep it in the presence of oxygen, that is with air and without air. Then getting all these answers, then if you have two organisms which have identical, which have, for example, I say the same round shape, both are gram positive, both are anaerobic, then to distinguish between them, we will look into the metabolic pathways, what is the type of sugar which it uses. So, like that we will do certain specific tests. So, for clinical identification, how do we identify? So, we will isolate the pure sample of the microorganism, you identify a single colony, plate it and grow it, and then you will do a series of biochemical tests finally to identify what is the organism which is present in the sample which is given. Then if it is identical organisms, then you have the serological tests. We use specific antisera to classify different bacterial species. So, same genus but different species. To identify those, we can use the serological tests. So, this is based on either the carbohydrate or the protein antigens which are present on the outer side of the bacterial cell wall, or which are present on the what we call as the polysaccharide wall outside. For example, you have specific tests for the streptococcal M proteins, or for Salmonella O and H strains you have specific traits to identify them. So, such final identification tests are based on what we call as the serological systems. When we look into the classification, as I said, based on shape, the cocci, the spiral and the bacilli. So, cocci are spherical, the bacilli will normally be very long and thin, and you have the in-between class what is called as coccobacilli, which is neither coccus nor a spirillum. And in some cases you will see curved and spiral bacilli with different wavelengths also. So, the shape in some cases are rigid, some organisms are in between. Then based on gram reaction, we call them as either gram positive or gram negative. As you see in this picture, this reaction is based on the difference in the thickness of the peptidoglycan cell wall. As you see in the picture, the gram negative ones have a smaller, thinner peptidoglycan layer, whereas the gram positive ones have a thicker peptidoglycan layer. Then based on the atmosphere, the growth atmosphere, now this actually we basically classify them as aerobes and anaerobes, but you do have a further subdivision here. You have what you call as obligate aerobes. These absolutely require oxygen for growth. Without oxygen they will not grow. Then among this you have what you call as the microaerophils. These do require oxygen, but they require a lesser amount of oxygen. So, they do not require the oxygen content what an obligate aerobe actually requires, but these microaerophils actually require lesser oxygen content. You have the obligate anaerobes, they require absolutely no oxygen. Then you have the facultative anaerobes. Okay, these can either be aerobic or anaerobic. They can shift their metabolism between both depending upon what the percentage of oxygen which is present in the growth area. Then finally, you have the capnophiles, which require a higher amount of carbon dioxide for growth. So, based on the type of growth atmosphere, we are now seeing 5 different types of classification of microorganisms. Now, there are some organisms which are capable of producing spores. What are spores? These spores are actually reproductive structures what are formed for existence when the organism faces what we call is reduced food nutrient condition, or when the atmosphere grows non-conducive for growth. So, then for survival they will grow into a space which is called a spore, where the nucleic acid and the material of the genetic material of the organism, and it goes into very less metabolic activity. So, all organisms, all microbacteria do not produce spores. So, based on whether they produce spores or do not produce spores, we call them as spore formers or non-spore formers. Then finally, based on the biochemistry, the metabolic pathways and genetic classification and serology typing, we can classify and come into a conclusion that the organism which you have seen or which you have, the sample is identified as this. So, this is the basic tests which are done to identify a microbe. In this session, we saw the general aspects about microbiology, how we classify the microbes, what are the different methods by which you can identify the organisms. In the subsequent lectures, I will be talking about the association of food and microbes, the foodborne illnesses, how do we identify whether a food is contaminated with microbes, what are the tests which are being done. Then when you are starting a food industry, then so pathogenic organisms should not be there in the food. So, what are the steps which you take for regulatory issues concerned with it? All these would be subsequently dealt with in the series of lectures. Thank you.